All right, cool. Guess I will begin now. All right, welcome everybody to another Use the Book Club meeting. Thank you all for coming. This week, I'll be going over membranes, and continuing the structure and function of cells lecture from last week. This lecture will mostly cover the membranes chapter in Campbell, and I believe there was another chapter assigned. So if you want to focus on that in your reading, that would be good. And I guess I'll just start lecturing now. All right, we're going to start off with a quick introduction. So membranes are pretty important because they surround and protect the cell and separate it from its surroundings. And this allows for the cell to regulate its internal environment. Plasma membranes are selectively permeable, which means that some substances pass more easily through them than others. The major component of membranes are phospholipids, which are amphipathic molecules with a hydrophilic head that faces outwards and a hydrophobic tail that faces inwards. The most common membrane phospholipid is phosphatidylcholine, which is composed of a polar head group made up of phosphate and choline, two fatty acid tails, and a glycerol backbone that joins these groups. So right here is the chemical structure of a phosphatidylcholine molecule. You can see the choline group right here, the phosphate group right here, the glycerol backbone, and the two fatty acid tails that are joined to the head group by the glycerol. So membrane phospholipids are structured in a bilayer in which a membrane is composed of a double layer of phospholipids, which you can see right here. The bilayer exposes the hydrophilic heads to water while sheltering the hydrophobic tails. And this forms a structure that is about 3 to 4 nanometers thick, which is the length of the two phospholipid molecules next to each other. Membranes also contain other components like membrane proteins and glycolipids, and the components of a membrane make up a mosaic consisting of proteins and carbohydrates surrounded by the fluid lipid bilayer, which is known as the fluid mosaic model. And with the addition of membrane proteins and other components, the membrane can be anywhere from 3 to 8 nanometers thick. Membranes are fluid because they're held together by weak hydrophobic interactions, and this allows them to move at speeds of around 2 microns per second. And right here, you can see another representation of a phospholipid with the tails and the heads. And I'll be going a bit more over the structure in the next slide. All right, so temperature changes and certain molecules can alter the structure of plasma membranes and thus change its fluidity. So membranes are more fluid at high temperatures and less fluid at lower temperatures. And in the lower temperatures, they're in a more tightly packed state. Unsaturated fats presented in membranes tend to increase the fluidity of the membrane. And this is because kinks in the fatty acid tails of these lipids, where the carbon-carbon double bonds are, tend to cause the tails to bend, meaning that they're packed together less closely, and thus they can be more fluid. And you can see that right here with there's fewer phospholipids per portion of membrane, which allows them to move around more and thus makes the membrane more fluid. And when you're considering saturated hydrocarbon tails, you can see that they're packed a lot more closely together, and this allows them to be, this does not allow them to move as much. So the sterile molecule cholesterol also causes structural changes in lipids. And a sterol is basically a nonpolar molecule with a characteristic 4-ring structure and an alcohol group at the 3rd carbon of the alpha ring. At higher temperatures, such as 37 degrees Celsius or body temperature, cholesterol interferes with the movement of phospholipids, and this makes the membrane less fluid at higher temperatures. But at the same time, cholesterol also stops the phospholipids from packing closely together, so it reduces the temperature necessary for membranes to solidify. So that means that cholesterol basically acts as a fluidity buffer, allowing for the membrane to remain at a constant fluidity. And going to consider the structure of sterols, this is basically a sterol. So you can see that there's a characteristic 4-ring structure with a hydroxyl, also known as an alcohol group at the end. And right here, you can see a representation of cholesterol inside the phospholipid bilayer, and you can see how it buffers the fluidity. And plant cells have low levels of cholesterol in comparison to animal cells, and instead rely on other sterols called phytosterols to buffer membrane fluidity. And bacterial cells rarely have sterols and usually use hopanoids, which are kind of a 5-carbon ring, which are 5-carbon ring compounds to buffer membrane fluidity. So that acts as like a cholesterol analog in the bacteria. All right, moving on, now I'm going to be talking about membrane proteins. So as I mentioned before, the cell membranes contain membrane proteins, and they're like a diverse array of proteins, and their structure helps determine their function. And the proteins themselves often determine the function of a cell membrane. So integral proteins are proteins that extend into the hydrophobic region of the lipid bilayer, and polytopic proteins, which are also known as transmembrane proteins, are a class of integral proteins that extend through the whole length of the lipid bilayer. On the other hand, monotopic proteins are another type of integral protein, which only span part of the bilayer, and they only stick out from the bilayer on one side. Hydrophobic regions of the integral proteins usually contain stretches of nonpolar amino acids that are around 20 to 30 amino acids long. So to put this into context, to span the 3 nanometer bilayer, an integral protein must be at minimum 20 amino acids long. The amino acids are arranged in structures known as alpha helices or sometimes beta barrels. Some integral proteins have hydrophilic channels that allow for the transport of substances through the membrane. And integral proteins can be dissociated from the membrane via a detergent, which has nonpolar and polar regions, which binds to the hydrophobic region of the protein and solubilizes it. So right here, you can see a representation of detergent binding to the monotopic or polytopic integral protein, and then removing it from the membrane, bringing it into solution. And this is often this is a technique that's often used by researchers to dissociate proteins from the membrane, and then they can then analyze those dissociated proteins. So on the other hand, peripheral proteins are proteins that are bound to the surface of the membrane, and they're bound to sites such as exposed integral proteins. Association with other membrane proteins occurs via hydrostatic interactions and hydrogen bonding. Peripheral proteins can be dissociated from the membrane through changing the pH or the concentration of ions, or direct removal, or the removal of calcium ions. So the removal of calcium ions causes dissociation of these and of the peripheral proteins because calcium ions stabilize peripheral proteins through ionic interactions with amino acids in these peripheral proteins. Specifically, they interact with the amino acids aspartate and glutamate. So adding a chelating agent, which removes calcium from the solution that the cell is present in, can dissociate the peripheral protein. And you can see an example of that right here. We're adding a chelating agent, which is urea or carbonate, and that causes the peripheral protein to dissociate from the integral protein that it's bound to. And other chelating agents such as EDTA or EGTA can also be used to dissociate these proteins. And likewise, as with the integral proteins, after dissociation, they can be analyzed for their amino acid content. So there's one last class of proteins, of membrane proteins, which are the amphitropic proteins. And these are membrane proteins that are only associated with the membrane some of the time. Association of the amphitropic protein to the membrane is regulated by changing its structure or its shape slightly. And membrane proteins facing the interior of the cell are held in place by attachment to the cytoskeleton. Proteins that are facing the exterior of the cell may attach themselves to extracellular materials, and attachment of proteins to other substances gives the cell a stronger framework. In addition to providing support via attachment, membrane proteins have functions such as transport, joining cells together, functioning in recognition, transducing signals through the cell, and acting as enzymes. Right here, you can see a representation of an amphitropic protein that's anchored to the cell membrane via a lipid and carbohydrate. And then right here, you can see a representation of the various functions of proteins, which include, like I said, the attachment to the extracellular matrix for support, both passive and active transport, joining to other cells, cellular recognition, acting as enzymes to bind to other molecules and help speed up reactions, and transducing signals through a cell from extracellular signaling molecules. All right, next I'll be talking about hydropathy plots. So the presence of hydrophobic regions of amino acids that are long enough to span a membrane as an alpha helix is a good indication that a protein is an integral membrane protein. And this is because long stretches of hydrophobic amino acids are rare in proteins that are not integral membrane proteins. So the hydrophobicity of amino acids is measured by experimenters by measuring the relative partition from water to a hydrophobic solvent, which gives a number to each amino acid known as its hydropathy index. So basically what happens is that they take water and then they take they take a hydrophobic solvent such as ethanol, and then they put amino acids in the water and ethanol, and they see like towards which solution the amino acids gravitate towards, and then those that strongly gravitate towards ethanol are given a certain number, and likewise those that gravitate strongly towards water are given another number. So more negative numbers refer to proteins that are more hydrophilic or water loving, and more positive numbers refer to proteins that are more hydrophobic. So right here you can see the hydropathy index values for but all of the 20 amino acids. You can see that isoleucine is very hydrophobic, while arginine is very hydrophilic. So from these values, you can construct a hydropathy plot. And how this is done is that the hydropathy index values for a window of amino acids in a protein sequence is summed and then averaged. And then the hydropathy values for the next window of amino acids is summed and averaged. And this continues for the whole sequence and allows for a plot of average hydropathy score to be made. So the reason that we're taking the averages for a window of amino acids, or just like a small sequence, instead of just plotting one hydropathy value at a time, is that if we plot one hydropathy value of like each individual amino acid in the sequence, then you're going to get a plot which has values that are all over the place. So for example, some are going to be like very hydrophilic, some are going to be very hydrophobic. And then the problem with that is that you can't really use that kind of plot to determine which sequences of a protein are most likely to be integral membrane proteins. And adding on to that, you can see that right here is a hydropathy index plot for the protein glycophorin. You can see right here that there's one hydrophobic region from these amino acid residue numbers. And on the bottom here, we have a hydropathy plot for bacteriorhodopsin, which has 7 different hydrophobic regions. And each of those seven regions is an alpha helix, since the protein, and this correlates to the 7 alpha helixes in the protein. And one last thing to add is that the window of amino acids should typically be a lot less than the length of the membrane protein. If the window is too big, then you risk missing certain regions of hydrophobic amino acids. Now I'll be moving on to membrane carbohydrates. So carbohydrates that are attached to the surface of membranes are often used in cell-cell recognition. Cellular recognition allows for the sorting of cells during development, and it also provides the basis of the immune system. So the binding of one cell to a carbohydrate on the surface of another allows for recognition. And membrane carbohydrates are short and branched, and they're typically 15 monomers or fewer. The majority of membrane carbohydrates are glycoproteins, which are just carbohydrates that are bound to membrane proteins. And there are also glycolipids, which are carbohydrate chains bound covalently to lipids. And an example of the carbohydrates on cells is the ABO blood type system, which is based on variation in the carbohydrate chains of glycoproteins on the surface of red blood cells. So the carbohydrate involved in the ABO blood type is built around a sugar core of 5 to 13 sugars, which is known as the H antigen. And the sugar core ends in a fucose, which is a hexose deoxy sugar. And after this, the O blood type has no additional modifications. In the A blood type, an extra sugar, which is N-acetylgalactosamine, is added to the end of the sugar core. And in the B blood type, the extra sugar is D-galactose. And this system allows for the recognition of the body's red blood cells and thus the recognition of foreign cells. So right here, you can see the structures of N-acetylgalactosamine, which is present in the A blood type, and D-galactose, which is present in the B blood type at the end. And the main reason that I included these structures is just to emphasize how much a small difference in molecular structure. So right here, you can see that both of these are almost identical, except for just one group at the end. So I want to emphasize how much a small difference in molecular structure can cause like a big change. And that's seen as how the A and B blood types are significantly different to the point that the body will reject A or B blood types, depending on what blood type it is. And the structure right here is just for L-fucose, which is the base sugar and is in the O blood type. And this diagram right here is just another representation of the carbohydrates in red blood cells. Right here, we can see the carbohydrates in the O blood type, the A blood type, and the B blood types. And you can see in addition to the sugars I talked about, there are a couple more sugars like glucose and N-acetylglucosamine, which are located on as part of the other parts of the other sugars on the carbohydrate chains. And these short carbohydrate chains are known as oligosaccharides. Okay, so now I'll be talking about membrane synthesis. So membrane synthesis is also the cause of membrane-sidedness. And the sides of a membrane are different because of differing lipid compositions on each side and due to the presence of different proteins and carbohydrates on each side. And membrane-sidedness arises as the membranes are built in the endoplasmic reticulum and the Golgi. So right here, you can see the process of membrane synthesis. So we start in the endoplasmic reticulum, and carbohydrates and transmembrane proteins are added to the membrane, and carbohydrate portions of the membrane can be later modified. Then after that, the glycoproteins and the membrane is then transported to the Golgi apparatus via a vesicle, and then in the Golgi, the glycoproteins will undergo further modifications to the carbohydrates, and the lipids will acquire carbohydrates at times and cause them to turn into glycolipids. And then the Golgi then transports the finished product in secretory vesicles to the plasma membrane. Vesicles will then fuse to the plasma membrane, and the outside face of the vesicle will become continuous with the inside of the cellular plasma membrane, while the inside of the vesicle will become continuous with the outside of the cell. So membranes have selective permeability, which means that the hydrophobic lipid bilayer allows smaller nonpolar molecules such as carbon dioxide, oxygen, and hydrocarbons to pass through easily. And the hydrophobicity of the fatty acid tails in the interior of the membrane impedes the movement of polar or charged molecules, which are hydrophilic. And larger molecules are also hindered from entry via just diffusion through the membrane. So therefore, transport proteins are used to shuttle molecules across the lipid bilayer, such as polar molecules, which would otherwise take a long time or be unable to travel through the lipid bilayer. Channel proteins are transport proteins with a hydrophilic channel that allows molecules to pass through. Aquaporins are a type of specialized channel protein that allows for the bulk flow of water into the cell, allowing for water molecules to enter cells at a rate of 3 billion molecules per second, which is a lot faster than just diffusion of the water molecules through the lipid membrane. Carrier proteins are transport proteins that change shape to allow molecules to be transported across the membrane, such as the glucose transporter. And these types of proteins are specific to only the molecule or group of molecule that they bind. And this specificity allows the glucose transporter in red blood cells to transport glucose 50,000 times faster than just diffusion of glucose through the membrane. So right here, I have a 3D view of an aquaporin protein. So this is from a site called RCSB PDB, or Protein Databank. You can just go to the site anytime you want, and then look up any protein you want to see in a 3D view to visualize it. And this site helps you observe how the structure of a protein will determine its function. So in the case of the aquaporin, we have a bunch of alpha helices spanning the membrane. And then when I go here and I add a molecular surface representation, wait for that to render in, you can see what the exterior of the protein looks like, the exterior surface, and you can see that channel there that allows for the diffusion of water molecules. All right, now I'll be talking about the different types of transport in more detail. So passive transport is a type of transport that does not require energy to perform. And passive transport is possible because of diffusion, which I mentioned before. Diffusion is the process in which particles of a substance spread out into available space. And the individual molecules of the substance move randomly, but diffusion in a group of molecules can be in a certain direction. Molecules will diffuse down their concentration gradient, which means that they'll diffuse from a higher concentration of molecules to a lower concentration of molecules. And passive diffusion includes simple diffusion through the membrane, which is seen right here down the concentration gradient, facilitated diffusion through a membrane protein, which is seen right here, and also with ion channels right here, and ionophore mediated ion transport, which uses molecules that bind reversibly to ions, and then these molecules then go into the membrane, deposit the ions, and then come back out, grab more ions. And this is also down the electrochemical gradient of the ions. So it's also passive. And then active transport uses energy to transport molecules against a concentration gradient. And this includes both primary and secondary active transport. In primary active transport, you go against the electrochemical gradient and is driven by the hydrolysis of ATP, which provides the energy to transport molecules. In secondary active transport, you go against the electrochemical gradient, but instead of hydrolyzing ATP to produce energy, you transport a second molecule along with the first molecule to be transported into the cell. And the transport of the second molecule outside of the cell allows for you to transport more molecules into the cell. All right, now for my last slide, I'll just be talking about bulk transport, which is another specific type of transport. So bulk transport includes exocytosis, which is the secretion of molecules by a cell through the fusion of a molecule with the lipid membrane. And exocytosis is used to transport large molecules like proteins and polysaccharides. And proteins rearrange the lipid molecules of the bilayer to allow the vesicle to fuse with the bilayer. And then once the vesicle fuses with the bilayer, it can then secrete molecules. And secretory cells use exocytosis to release hormones and neurotransmitters. On the other hand, endocytosis is the uptake of molecules through the formation of new vesicles from the plasma membrane. The plasma membrane forms a pocket that pinches in with the substance to be uptaken inside the pocket, and then that then forms a vesicle with the molecule that's outside of the cell now inside the cell. And some of the types of endocytosis include phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, you can see that the cell will engulf a large particle by extending itself outwards. In pinocytosis, the cell will pinch inwards to allow molecules to flow into that pocket, you see, and then it will then pinch the membrane again to form a vesicle that will go inside the cell. Lastly, in receptor-mediated endocytosis, the cell will again pinch inwards, but this time there'll be receptor proteins on the exterior surface of the cell that will attach themselves to solutes to be uptaken into the cell. And this allows for the cell to uptake specific solutes instead of just taking a bunch of solutes all in at once. And that's pretty much the end of the lecture part. I've got a quick Mentimeter which is just a type of quiz for you guys to play. Let me load that up. So just go to menti.com and enter this code. Then once a couple people join, I'll start. I'll give it another couple of seconds and no one else joins, then I'll just go ahead. All right, is everyone who wants to join joined now? I guess if anyone has not joined, then just speak out. If not, I'll just start. All right, I guess we'll just go ahead and start right now. All right, this is a somewhat time-consuming question, so there's quite a bit of time. So yeah, just take your time on this. You'll need information from last week's lecture to get this question. So if you don't get it, just don't worry. You can always go back to that lecture, watch it again, or check out the slides. All right, yeah, so the correct answer to this was this choice right here. So I guess we'll just go over why. So with the first, when you're looking at a transmembrane protein, uh, you're you'll be looking at something that is 20 to 30 amino acids long and is largely hydrophobic with hydrophobic residues. Uh, so with other three choices, you can see that there's a lot of hydrophilic residues present. With this choice, you have threonine, serine, the arginines, a lot of glutamines, which are all hydrophilic and are thus not likely to be present in the transmembrane domain. Right here, you have arginine, asparaginine, aspartate, uh, and a bunch of couple other hydrophilic residues. So once again, that's not likely to be present in transmembrane domain. And right here, you also have a decent amount of hydrophilic residues, including all of these histidines, the proline, the asparagine, the arginine. So that's once again not too likely to be present. And this choice contains all hydrophobic residues, which means that it is most likely to be in a transmembrane domain because it can be present inside the lipid bilayer because it's hydrophobic, and the fatty acid tails of the lipid bilayer are also hydrophobic. All right, moving on. Right, most people got this correct. So let me go over the question now. So question talked about you're studying a hepatocyte membrane. So this is just a liver cell, and you find a protein that transports fructose into the cell. The specific name of the protein, I believe, is called the GLUT2 receptor. And we want to know which of these terms best describes the protein. So with the peripheral protein, that's not super likely since it's just attached to the outside of the cell. Uh, with the monotopic protein, it only spans part of the membrane, so you can only bring glucose in part of the way. With the polytopic protein, it spans the entire protein in the entire membrane, so you can bring the sugar the whole way through the cell and into the cell. And with the amphitropic protein, it's only attached to the cell part of the time, so and it doesn't span the whole bilayer of the cell, so it's also unlikely to be able to bring fructose in the whole way. All right, once again, the majority of people got this correct, or my bad, did not get this correct. So I'll go over this. So with the hydropathy plot, you want to choose a window of amino acids that is shorter, that is significantly shorter than the the length of your protein sequence. And that's because if you choose a sequence that is too long, then uh, your your protein sequence will likely get lost when you average out the values. So with the 40 and 30 sequences, they're much longer than the 25 amino acids. So you can eliminate them immediately. With the 20 amino acid sequence, it is shorter than 25 amino acids, but it's still quite close to that length. So you also want to eliminate that. And that leaves the 10 amino acid sequence, which is short enough that you will not likely miss your 25 amino acid long integral membrane protein sequence. Sorry, these answer choices are a bit small. Try to zoom in a bit. All right, yeah, so this answer right here was the correct answer. The other answers, starting off with this, xylose is a plant sugar, so it's just like not present in humans, I believe. This one right here is N-acetyl galactosamine, and same with this answer right here. And the problem with that is that N-acetylgalactosamine is only present in the A type of carbohydrate. So you can eliminate those two answers, and that just leaves this answer right here, fucose, glucose, and N-acetylglucosamine. And that's all the questions I have. And yeah, thank you for participating, everyone. All right, you can see I have a couple of questions right here. Let me try to answer them. All right, someone's asking me to go over monotopic integral proteins and polytopic integral proteins. So I guess I'll go back to that slide. Just give me a couple seconds. All right, here it is. So with the monotopic protein, you can see that it only spans part way into the bilayer, and it only sticks out on one side of the membrane. Meanwhile, with the polytopic membrane protein, you can see that it spans the entire bilayer and it sticks out on both sides. And that's pretty much the difference, just the length that they span. All right, I have another question about the receptor-mediated endocytosis. So I think asked about like how efficient, why would it be more efficient than just bringing them bringing the things in indiscriminately by normal endocytosis. So I think the answer to that is as described in the question, that it just is targeted to certain areas of the cell. So it's generally designed to bring in specific molecules. And I think that's all the questions that I received. Can we see the leaderboard? All right, I think there's the leaderboard to this. Let me try to pull it up. All right, congratulations to this person for placing the highest. And yeah, that's pretty much it for this lecture, unless anyone else has more questions. All right, yeah, thank you all for coming. I guess since nobody else has any more questions, y'all can just leave if you want, or you can stay and ask questions. And yeah, thank you so much for attending.